Hi, everyone. Welcome to Examining Evolution. I'm here today with two good friends, Michlas, the Danish debater, and Sal Cordova. And Sal is a molecular biophysics researcher, and he's going to be talking to us today about zinc finger proteins. And so we called this giving evolutionary biologists the zinc finger. And that is because what Sal is going to be doing today is showing the complexity of the zinc finger and how hard it would be for this to evolve by natural processes. So welcome, guys. And then Michlas is here because he's a great thinker and question asker, and he's going to be kind of helping the conversation flow by asking Sal good questions. So glad you guys are here.
Well, thank you.
Thank you.
I hope I can live up to that.
Well, I'm here at the service of Rebekah and Michlas. This isn't going to be just a data dump lecture. I have given parts of my presentation were synthesized from several lectures I gave to Christian faculty, science faculty, and researchers at both Christian institutions, secular institutions, and the NIH. And I got good reviews on this. In fact, one of the deans of one of the universities, after he saw this lecture, started taking slides from my slideshow and sharing it with his students in his biology class. So I was very flattered by that. My purpose in doing this really is just to show angles of the complexity of biology that aren't discussed very much in terms of the difficulty of evolution. I think right now I might be the only one that is exploring this. I would hope that whichever side anyone is on this issue, that they might feel they've been able to come away with this, just learning a little bit more about biology and the wonders of life. And then we can all feel like time was well spent and we can come away feeling good. So that is really my goal for today. That's in addition to me just arguing the improbability of all this. So thank you.
Thank you. And Sal has a YouTube channel called Evidence and Reasons, and also Evidence and Reasons Academy. And did I say it right? Is that the right?
Yes.
Okay. And so the link to his YouTube channels are in the description. So please subscribe to him if you enjoy this content. Well, we're ready for the zinc fingers, Sal. Go for it.
Okay, if you can bring up the slides. All right, let me make sure I have it here. All right, when your zinc fingers aren't working, you might suffer brain damage. And this was just recent scientific reports, like, for example, the loss of zinc finger proteins in Purkinje cells can be damaging. They'd knocked out these zinc finger proteins and also subjected these poor mice to alcohol, and it damaged. It killed the Purkinje cells. They're supposed to, there it is. There's a picture of the Purkinje cells. Purkinje, he's Czechoslovakian. And there's a picture there of a mouse Purkinje cell. I'm presuming the poor thing was decapitated so they could extract the poor cell. I shouldn't laugh at that. The zinc finger protein is very critical to the operation of things like this and so many other things, including the liver and all sorts of cells. And you'll start to see why in this lecture. And what they've been doing to these poor creatures is they'll disrupt the zinc finger in the mouse, either by like gene knockouts or chemicals. And we've associated the loss of zinc fingers with the loss of these cells, the atrophy of these cells, and the death of these cells, and the dysfunction of these cells. And it does lead to human issues if your zinc fingers aren't working. So scientifically, we can only establish improbability. The mechanism of overcoming improbability might be outside experimental, repeatable science. People who know me know that I think the improbability suggests God, but that's not formally provable. Some people will just say we somehow life beat the odds and we're here, no need of an explanation. Some people just invoke an unknown mechanism, and some will invoke space aliens. We're not going to go there today. We're just going to explore, we're just going to see the complexity of biology, and then you'll start to have a feel for the improbability. So this is just some basic biology here. We have proteins that are coded by DNA, and the protein can be spelled, it can be represented with English alphabetical letters or abbreviations. And often a protein, technically we could call it a polypeptide, it's just this long string of hundreds of amino acids. Any problem there? And we can represent it with spelling in alphabetical letters. And I'll do that right here. Oh, wait. Anyway, these are the amino acids. That's the molecular diagram for completeness. And as I said, you can represent the amino acids with English alphabetical letters, and they can also be grouped like according to this Venn diagram in terms of their properties. This is one of my favorite examples. If you just take that string and you just kind of make it like a paragraph with wraparound, this is the collagen. Collagen, you manufacture collagen. You can actually buy collagen supplements to make you look beautiful. And this is like one of my favorite examples. You could actually see a non-random pattern in the collagen spelling. And that's a signature pattern. You can identify collagen just by that that's a collagen. If you're looking in the cell and you see these proteins or the genes that code for it. I mean, we can also go to the zinc finger. This is the spelling of a zinc finger. You can see it's shorter than the collagen, about 1/3 the size. And it also has non-random patterns here, and I'll explain the significance of that. It's very important that these non-random patterns are there. It affects the biophysical processes. And as I said, with the Venn diagram, you can group the kinds of amino acids. You can have these higher order patterns that are non-random. And it's just brutally evident on this particular zinc finger protein. You can't do this on most proteins, but I picked out two where visually you could see the non-random patterns. And this is called a classical zinc finger. And I'm going to just keep pushing forward. I'm just going to pause now. Are there any questions about kind of this that may pique your interest before moving on, just so we all don't fall asleep? I like being interrupted. Okay.
No, just listening right now.
All right. And some of you know that I presented this at the CRS, why you could see there's not going to be a common ancestor between, like, say, a collagen and a zinc finger. Even Dr. Dan would agree with me. That's different than the question of universal common ancestry, but in terms of a universal gene or protein, both he and I agree that we believe in independent lineages for the protein/gene. Technically speaking, it's the locus. That's neither here nor there. So I'm going to just, rather than define what a domain is, I'd rather kind of just show you a domain, and then I'll formally define it, just so you could kind of get the notion. So here is the zinc finger. See if you can see my cursor. Good. So here is the zinc finger spelling here, and you could see each line there. We call that a domain. Each line is a domain, a zinc finger domain. And if we wanted to represent the fold with a Richardson ribbon diagram, if we took one of those domains, the way it would look, if we just snipped it out of the protein, it would fold like that. We have the zinc ion here, and it's connecting to the C's and H's. So you could see the C's in red and the H's in purple. It connects. A two-dimensional approximation of that is this fold. So this is a more high fidelity type fold. It's a Richardson ribbon diagram. It uses ribbons to represent the molecules. It's very, the reasons that they do it that way. Now, what I'd like to emphasize is, just like you have the architecture of a key, and each of the indentations is different on each key, but you still have the same basic architecture. You could see that each line is spelled slightly differently, but it has still the same basic architecture of the C's and H's in a given position. That's very important. It's very much like this key. It has to have certain rules for parts of it, and then parts that are variable. That's very critical to this. So I'm going to kind of appeal to a kind of a lock and key analogy here. That's not exactly some people cringe at that, but that's, in the everyday world, that's what we can most relate to. So just think of spelling as kind of either changing the lock or the key. And I'll just keep moving forward, and please interrupt me. I'm going to look for you.
Yeah.
Oh, finger.
Yeah.
Michlas.
So can you go back just the other slide? We have all these colors. So we have each zinc finger here. For each of those, which one is the, should we say, the grooves on the key, those that actually have the code? What part of this zinc finger is it that has to have a sequence that match something or represent a matching sequence?
I have diagrams that will show that later, and that might be, I don't know what sequence. Let me see if I could show it. Can you hold that thought? I'm going to try to answer that question by just moving forward. And I'm really glad, because you're kind of on the wavelength I'm hoping you were. You're asking these questions. So just a little technical stuff before we move to actually answering the question. So this is just for graphical purposes. So you can see each line, each row here in the spelling represents an individual zinc finger, and there has to be a zinc ion in each of these. So there's a reason I'm kind of drawing this out, because you'll see how it looks a little bit more like a lock or a key, or password and login. I'm sure someone out there can think of a better analogy, but again, let me just emphasize that these are individual domains. See, okay, so this one emphasizes the spelling. If you look carefully, the spelling, you can see the spelling here in between is different. So just look carefully in this region here. Let me see if I could use my laser pointer. Oh, my laser pointer's not working. Hang on. No, I can't use my laser pointer, guys. Sorry. But you could see the spelling in that region is different. So that would kind of correspond to the indentations you might see in the key, or the tumblers in the lock. All right, so the reason they called it the zinc finger is that it grabs, one of the functions of the zinc finger is to grab DNA. So get this, DNA codes for a protein called a zinc finger, and then zinc finger proteins will actually grab the DNA. And occasionally, I don't know if we ever have a circular loop where the zinc finger grabs its own DNA. That's an outstanding question. But, okay, so you could see here, this is kind of the diagram. So I'd say the stuff in between actually the whole spelling, but the differences in spelling here, where you see it's not highlighted with colors, that's kind of important to you might say those are the indentations. Let me show a particular, this is another zinc finger, but you can see it doesn't have quite that nice lined up thing where, I had to insert spaces to get things to align. The way we know that it actually is a zinc finger is when the protein is expressed, we can say that the zinc ion will actually connect to these Cs. That's how we know that it really is a zinc finger. So sometimes we get lucky where the spelling is so clear, we can use automated algorithms that just check the spelling and say this is a zinc finger protein. Other times it's going to be a little bit more difficult. We can kind of guess. The computer might say it might be a zinc finger, suspect it, but the way you confirm it is to actually, see at the molecular level, see if zinc ions connect to these locations. So again, the zinc would have to connect to the C and the H, and then another zinc ion here. So this is the transcription factor 3A. And so you can imagine here, this is the zinc finger protein, and then it's connecting to a stretch of DNA. Now this is not to scale. This is the zinc finger connecting to about 120 long stretch, 120 nucleotide long stretch of DNA. But you could see kind of the password login analogy or lock and key analogy. The spelling of the zinc finger here, in terms of its letters, will specifically identify the DNA it's supposed to connect to. And this raises as we started to build man-made zinc fingers, in the man-made zinc fingers, we every 3 DNA bases would require a separate zinc finger. In this particular one, it's not. Michlas, please.
Yeah, so just back to that one you showed, that all the zinc fingers are the domains connecting up to the one we had just before.
Before it?
No. Forward. One more,
okay,
I think.
So you see this line here, and then I'm going to go down. Is this the one you want?
That one. Yeah, that one.
Okay.
So here we see what the 11 zinc fingers domain connecting to give you this 120 long connection. How many, if you have a zinc finger protein with 11 zinc fingers, I think, was what you had before, I think. How many of those will connect to the DNA? Will the all 11 connect, or some of them to some DNA or some to others? And this ties into what I have read so far. It seems like they don't always all connect. We usually have something like 3 or maybe 4 at extreme cases.
It depends on the particular protein. That's a very good question. So the man-made ones, I said one zinc finger targets approximately 3 DNA. That's the man-made ones, because that's the best we could do.
That's the finger itself. I'm talking about how many fingers will target the DNA on one zinc finger protein.
It depends on which one, because some are very specific, what we would call, kind of loosely, we call it with monovalent. Mm-hmm. If it's going to target something this specific, that's a specific stretch of 120 long, all of them usually are involved. The multivalent ones, it'll modulate, and I may show a video of one that is multivalent, where sometimes it's just going to use the center, like 3 or 4, and then occasionally use the ones on the end, on this either left or right of it. Mm-hmm. So
now, is that you're saying it depends which one? Is it depends which zinc finger protein? Like, are there multiple types, or are you saying it depends which stretch of DNA, or what? I'm kind of confused.
Good question. There are multiple different kinds of zinc fingers, and they usually have a numbering system for which ones they identified. So there are multiple proteins that have zinc fingers, and some are just called zinc finger proteins. But there are other proteins that aren't called zinc finger proteins that are also zinc finger proteins. The way we know it, like this one's called the transcription factor 3A, but it has a zinc finger in it. So it's not called zinc finger something, but it is a zinc finger protein because it has these zinc fingers. And it's not the thing that will decide that it's a zinc finger is the fact that zinc ions will connect to the C's, the cysteines, and the H's, the histidines. So they're numerous. In fact, I think 1% to 3% of the human genome are made of zinc finger proteins. So many proteins have zinc fingers in them. So that's a huge number of proteins. Out of 20,000 proteins, so 1%, what would that be? Like 1 to 3%, 200 to 600 proteins in the human genome are zinc finger proteins. And each of them behave a little differently. So
do they all have what you call it, DNA targets? So if you have so we have some that does not have any actual targets to work with.
Some of them have RNA targets, and some of them have protein. Okay. Most of the time, we haven't characterized all of them. In fact, characterizing how a protein functions is actually really difficult. The experimental, the experiments that have to be run are just gruesome. Like, for example, I worked on the topoisomerase, and we have people that have spent their professional life on topoisomerases, and we have hundreds of those researchers, and we still don't understand. So this is very difficult to characterize it. We do know we have characterized some, And maybe those are the ones that I'm covering because they're very, even the researchers that have studied them, like this transcription factor 3A, the quotations are like, they're just amazed how well this is put together, how difficult it is. And this particular transcription factor 3A zinc finger can connect both to DNA, and then when the DNA is expressed as an RNA, the protein's able to connect to that. And the researchers were just, their jaw dropped. They said, that doesn't seem, you could tell almost that they're like, that's incredible. They use the word specifically sophisticated. It's multi-purpose. They're quite astonished. So thank you for asking. These are great questions.
Yeah, it's because I'm wondering, and that will of course tie into later considerations. If we have any zinc fingers, individual zinc fingers that we know do not attach to anything, or do we have any zinc finger proteins we know do not attach to anything?
I'm not aware of any. I'm honestly not aware of any. So thank you. And that is a good question, because I've challenged evolutionary biologists. I said, if you're thinking these evolve, you ought to, and you're using all these generous numbers of all the trials and whatever, and the waiting time problem that my boss John Sanford worked on. It's like you just don't see a lot of these that are swimming around that have no connection.
Yeah, because that's what I'm thinking. You don't absolutely have to have it for evolution, basically. But if you have it, that would be an indicator of parts of the pathway. I won't say a pathway, but part of it. So
exactly.
It's one where we can find a positive in this case.
Okay.
Or we could search for one, as it's called. Yeah.
Well, I'm yeah. Let me move on a little bit, and please interrupt me if I, hopefully this will trigger even more interesting questions. So you could see there, just think the difficulty. I'm going to see if I have something here that I wanted to show. Okay, good. I have. I'll show you another. There's a subtlety here that is very important, and I'm about to address it. So how does this relate to, we had this started in our talk on ERVs. One of the targets of zinc finger proteins are ERVs, but also other sorts of DNAs. So ERV, the DNA ERV, the zinc finger can bind to that and target it. In the diagram here, it's targeting something called 5S DNA. They use, that's a very famous stretch of DNA. And there are all sorts of things it can target. It can target large parts of the genome, and I'll show some of the things that it targets, things like that they call CTCF binding sites. There are all sorts of things. And I'm going to cover a little bit of the intricacy of all the, just attaching attaching a zinc finger to DNA, there has to be a lot more to make it actually useful, because otherwise it's just going to be like, a random piece of tape or glue just stuck somewhere, and then it doesn't do anything. So this is showing how a zinc finger connects this molecular complex here. And again, there has to be spelling here to make all these connections. And then the zinc finger is an intermediary between this molecular complex and the DNA. And that DNA could be an ERV. It could be lots of other targets. So we make man-made zinc fingers because we're trying to make therapeutic drugs or treatments. And sometimes it's nice to be able to try to make a molecular machine, like say we're trying to treat Alzheimer's, and we're trying to disrupt what may be making a bad protein. I'm aware of it. Someone had told me how this works, and I'm foggy on the details. But one of the applications is, what we would do is we would change the spelling of the zinc finger, and it's roughly about 32 amino acids long. So just going back here, this is about 28 to 32 amino acids long. 28 to 32 amino acids long. And in the man-made system, you would change that spelling of 28 to 32 amino acids, and then it could connect to 3 specific DNAs. And it's a little more subtle than that, because it's actually 4, because there's overlap. So this one has to spell like CCG and then an A down here. And then this one has to spell TGGC. There's overlap. Don't need to go there, but I'm not saying it. This is starting to show this is not trivial. We have great difficulty in building this because we sort of have AI to help us decide what the spelling should be, but it doesn't work as well as the ones we find in nature. Michlas.
Yes, sorry.
Oh no, this is good.
I stopped. You said what? 26 amino acids are translated to these 3 to 4 letters.
28 to 32 amino
28.
acids. Yeah.
28 to 32. So this means we have a lot of letters representing a few letters. Do we have multiple spellings for GC, TA, or are we limited to one specific spelling?
Multiple spellings. Multiple spellings. But there's going to be an interesting twist to this that I need to show.
Mm-hmm.
It has to deal with the fine-tuning of affinity, because not only does it target there, it has to connect with a certain right amount of strength, like a certain amount of glue. And I'll show you the problem. These You have great questions here. Your instincts are good. So I'm just showing kind of the references here if anyone wants to see it. So the man-made, we have kind of this model that I just showed you, the transcription factor 3A. It obviously doesn't hold to that model. Okay, so the man-made model, when I gave the three letters of DNA, that's the man-made model. The ones in nature can deviate from that. So this is the problem of fine-tuning of affinity. So we have like tape that has different strengths of connectivity, so to speak. Like if I'm painting my house, or a painter is painting something, they have masking tape. You don't want the tape to be too adhesive. You want to be able to strip it off very easily after you're done painting. Whereas sometimes for packaging, you want really strong tape. And the zinc finger here, to make a good zinc finger, it has to have the right amount of strength. And I'll show you why that's going to be problematic if you don't have it. So let's say, for example, you have this zinc finger and you spelled it, but it creates too much affinity. So you asked the good question, can you have numerous kinds of spellings of amino acids target a piece of DNA? Answer is yes. Is it a smaller set of that gives you the right affinity? Because, for example, let's say you have one that is like super glue. I'll just call it the super glue spelling. What's going to happen is this zinc finger is just floating around inside the cell, bumping around. That's how it does its search algorithm. It's using, figuratively speaking, Brownian motion. And it's going to bump, and if it hits a target that it doesn't really connect to, it's just going to move on. When it hits a target and all the connections are there, it's going to adhere. What is for it to hit and then, kind of release, what you want is that this is kind of more like painter's tape. It's not going to adhere too strong, because otherwise, if you have one part that is like super glue, it's going to be stuck to a position it's not supposed to. It'll basically override. It'll stick to that part of DNA, and it's going to override these guys may disconnect, but then this thing is so strong. And this is a problem in biophysics. My jaw kind of dropped when they're talking about the dwell time of, how long does it interact before it disses the partner and looks for someone else, looks for its true partner. And this is another reason why man-made zinc fingers are so hard to make. They have to deal with this sort of stuff. So have we calculated exactly the probabilities? Rigorously, no. If we were able to, we'd actually be able to make very good zinc fingers, which we're not. And so at this point, I can only give you kind of an intuitive feel of how difficult the problem of making a zinc finger that can target an array this large. It has to have, you can solve the targeting problem somewhat and say, okay, well, we can estimate how improbable the spelling is just to get it to the right sequence of DNA. But then from that, it's even more improbable to get one that has the right stickiness, the affinity. So Now I'll describe I'll formalize when I use the word domain. A protein domain is any identifiable subsequence of a protein that can fold, function, and exist independently of the rest of the protein chain or structure. And we kind of saw illustrations of that. I say protein domain is a definable component of a protein. Domains are somewhat quantized entities. So we have we have protein motifs. They're kind of general spellings, but a protein domain is kind of a little bit more of a functional unit. We also have DNA motifs, and those are spellings. So what you want is a protein domain that will look at when we have this connection here. So we have these protein domains, which are the zinc finger domains, and then you have this. I'm sorry, I don't know why it's doing it. Then you have this motif. This is a DNA motif or binding target that it looks for. So that's just a little bit of the terminology. I got interested in this a little bit. My professor at the NIH, Igor Rogozin, he's here. He works for Eugene Koonin. And he wrote this paper, Evolution of Domain Promiscuity in Eukaryotes. It's problematic that we see these domains kind of violating common ancestry. You kind of see it in one protein and another, particularly zinc fingers. Talk about that. So just for completeness, there are other kinds of zinc fingers. I'm talking about the classical one. Now let me show you a little bit more of the architecture of this 136 protein. This is a crab domain here, and these are the zinc finger domains. So we have 13 or so, and then a crab domain. And we can represent this also. That's what the crab domain would look like when it folds. And we can represent this with a diagram like, let me skip this. So, okay, this is an approximation. I don't have enough. This is like a block diagram for each row. I don't have enough blocks, if you'll forgive me. But and then we have a crab here. So this is the crab domain in the blocks. So sometimes the way we'll represent the protein is with these block diagrams. Here, I'm just going to highlight. This is the zinc finger, the crab zinc finger protein, and there are other kinds. We can people that hear about alternative splicing, the genome can express it with the crab and then the zinc finger, or sometimes just with the zinc finger itself. Now, this is an interesting one where the whole crab zinc finger complex will bind to the DNA, and then at some point it's going to split itself, and then this part interacts with the RNA. This is really sophisticated stuff. I mean, that made my jaw drop. People who understand the difficulty of this will really appreciate that. There are alternate ways to implement. We have different domains. We have crab A, crab B, and we can exploit this by mixing and matching one part with another. We can pull this out and make kind of like Legos and mix it for man-made applications. But we actually see this happen in nature, and I'll talk about that. So you can sometimes see, here, for example, you can rearrange. You'll see it in living organisms. You'll have the zinc finger like this, and then other times you'll have it kind of spread out with other Lego components. And this is relating to that paper by my professor, The Problem of Promiscuous Domains. If we take the block diagram to look at other different kinds of domains, you can definitely see how it shuffles differently between proteins. And so this is like the little kids with Legos. And it's really funny when I look at these protein databases, it reminds me of the kids with Legos because of the colors. Oh, Michlas, I'm sorry. I saw something in the private chat. It said, could you explain the consequences? Are of it being too strong or weak, or is it terminal or very effect? It could be terminal, meaning it could be just stuck there and you can't remove it. So let me just briefly show what would happen if you can't disassemble the machine. If it is, a lot of stuff in the cell has to be dynamic. meaning the molecular machines will visit something, do its business, and then be released and disassembled. If it sticks too hard, it could kill. It could end up repressing or blocking any future function. And actually, that is sometimes a medical treatment. If you want to just block something, you could park a zinc finger here that's just going to just stay there and never get removed. So sometimes that terminal behavior is used by medical researchers. But if we have something that's a bad zinc finger, you could see how this could disrupt all the flow of the kind of the factory of the cell. So it could be terminal, not necessarily. But the reason you don't want it to connect too strongly, and we'll see this later on, is after this is done doing its thing here, it's going to be modifying. It's going to be flipping bits on this chromatin computer here. This is what this machine will do. And we can go into that. After it's done, this is like a read or write head. It has to release. It releases, and then other read or write machines will come. So a lot of people don't realize this is a computational machine. This is like random access memory with multiple read and write heads. And so this is why it's very important we have the fine tuning of the stronger, the strong and weak interactions. So going back to this for the promiscuous domains, thank you all for your forbearance. I just love pretty pictures, and this is cute. So the proteins are made of all these modular components, particularly eukaryotic proteins. And there's this CDART database where you'll really see this, and you'll see kind of the Lego diagrams. And this is actually an argument, again, against common descent of all proteins from one ancestor, because it would have to be lots of gene splicing where you take parts of one gene and take another part of another gene and you splice them together to make a new one. And this is an example of it right there. These are diagrams I got from peer-reviewed papers that show all this kind of the gene splicing that would have to go on naturally. And I've objected to evolutionary biologists kind of waving this away as not a big problem. And just like, well, that was the point of my professor's paper. It's like, we don't know how it can do this. This is not just point mutations. You'd have to do translocation mutations where you take pieces of one gene and insert it in another in the right place. Anyway, oh yeah, so yeah, you could hear the various peer-reviewed papers had different representations for this SH3 domain, and you could see how it's laid out differently on different proteins. But anyway, let's move on because we're focusing specifically on the zinc finger protein. That was
could I quickly ask here?
Yes, please.
So not a common descent for all proteins, but what about all zinc fingers? Are they considered to have somewhat related descent?
How about we assume it for the sake of argument? I actually don't know what the evolutionists say. A lot of them that I've interacted will say, yeah, it descended from one. I don't think anyone would know.
That's a huge part if you want to calculate the probabilities later. I mean,
right.
That would be huge in that.
As far as my argument, I'm going to assume common ancestry from a zinc finger. And actually, you briefly saw a diagram where I did just that. There it is. So what I did is I took the zinc finger domains, the spelling. See if I had it. Sorry for paging through this. I had the wrong. Apologies, everyone. So, for the sake of argument, I'm going to assume common ancestry, and I had a picture here. Okay, so if I assume common ancestry, what I did is I took the spelling of each of these domains here. Let's see if I have it. Did I lose my mouse? Ooh. Okay, I take the spelling of each domain here, I put it in a phylogenetic software, and it can build a tree, and it looks like common descent, just like you'd suggested. But the problem is we would reconstruct the history, and you'd have these zinc fingers in different positions. And what I am arguing is, if you get the spelling wrong, the zinc finger is dead. It's functionless. At best, you hope it just goes to a neutral spot on the genome and doesn't cause damage. But if it's kind of a neutral effect, natural selection isn't going to kind of bring it closer to the promoter site or whatever the target it is. So yes, it would. I'm going to absolutely concede that you're right. It would reduce the probabilities. And so for the sake of argument, I will grant that. I said that's a legitimate possibility. They had said in that text debate that I sent you at Skeptical Zone, they're saying, Sal, you're so stupid. This is so easy by gene duplication. I'm just like, no, it's not that easy. First off, what if you cut it in the wrong spot? I mean, if you just randomly take a piece here and then it gets duplicated, you're going to destroy it. You're not going to be able to make a nice array. Building an array like this, it's not like the DNA is going to get cut in the right spot so that it nicely makes another zinc finger here. And they never worked through that.
But Sal, we've got billions of years. Don't you think out of billions of years, then those, things could just happen in the right way at least once?
At least once.
Let's just suppose it happens at least once.
But then comes the question: what would happen if, let's say, they copied it in the middle of a domain? Just middle of third domain copied into the middle of the fifth domain. Just
right. So the first thing is the C's here, in the red,
mm-hmm.
and the purples, they have to be spaced apart.
Yeah. I get that the zinc finger itself would not work, but what would be the consequences for the organism it happens in?
Well, that's why I showed, like, at the beginning, what happens when they disable, like, the zinc finger 212. Now, granted, they knocked out the whole gene, but I'm presuming you start to knock that out, you're going to affect the binding affinity. It may not even bind, and you end up just doing all sorts of damage. But rather than speculate, if anyone really wanted to, they could run the experiment and find out. But because we're doing synthetic biology with man-made zinc fingers, we're finding, oh my goodness, it's really hard to get it just right, because even with all of our technology, we're not making very good zinc fingers ourselves. We have to do a lot of trial and error, even with all our artificial intelligence and computation and whatever. So to Rebecca's question, it's like, for millions of years, like, if I had 500 pennies, I could have a child and he could make it all heads. But that would be longer than the age of the universe to try to do that by random chance. And natural selection is not available because something that is not working is probably not going to be selected for. And so we can just develop all these theories. And I appreciate these questions. But if I could just show more of the complexity, because I don't want to limit the discussion to just getting some of this, some of the parts of the zinc finger. It's important to see the context to how much else the cell has to have in place for the zinc finger to be useful. It's more than just, like I said, if you just had a zinc finger and it just binded to the DNA, that's just like taking glue and slapping it on the DNA, and it doesn't do anything. It only becomes functional if you have all these other parts. In the this CAP1 zinc finger complex is connected to the regulation of ERV. I mean, it interacts with ERV in some way. I'm not, there you have it. This is the reference that talks about the zinc finger protein here. And this is in 2019. I talked about the transcription factor 3A. That's the one I showed pictures of earlier. And see, they use the word sophisticated, the sophisticated zinc finger protein. And I'll just see this whenever they talk about zinc fingers. It's like, they're like, wow. So let me show another thing. I hope this video doesn't blow apart. I'm going to first take down this video. I'm going to stop sharing this screen. I'm going to share, hang hang on, let me get this going. I'm going to share another aspect of this, and I'll do a share screen. I'm going to have to cut my audio. You all may have to cut your audio too if you start getting feedback.
Yeah, we're muted.
Okay.
And sorry for looking so much away. I'm also trying to follow on in the chat if some good questions or something come up.
Right. So this zinc, you're going to see here something called the CTCF binding site. So the CTCF binding site is DNA. It's DNA. And then you'll see, I think, CTCF. CTCF is a zinc finger. And the target of the CTCF is the CTCF binding site. So try to get the terms squared away. You have CTCF, and then the thing it connects to, that's a DNA stretch. And let me just show this, just a minute of it. And I'll show the significance of this.
In a recent study, we show that when the 2-meter-long human genome folds inside the microscopic nucleus of a cell, it forms 10,000 loops. Each of these loops connects 2 anchor points which are far apart along the chromosome, but which loop back, coming close together as the genome folds. In our new study, we find that loops form by a process called extrusion. Let's take a look at how this operates on a segment of DNA. First, an extrusion complex containing 2 subunits attaches to DNA, forming a small loop in the process. Next, the 2 subunits slide along DNA in opposite directions, making the loop grow bigger and bigger. The extrusion complex looks for a specific motif, a DNA word, which causes a protein called CTCF to bind to the DNA. If a subunit encounters the CTCF motif that is pointing at it, the subunit tends to stop. If the CTCF motif is pointing the other direction, the motif won't be recognized. To the subunit, the motif looks as though it is backwards. This behavior leads to a result we call the convergent rule. The pair of CTCF motifs at the ends of a loop must be pointing towards one another on the DNA. Our simulations also show that when loops form by extrusion, they lead naturally to the formation of what we call loop domains.
Okay, I'm going to show. Hang on. Sorry, I pressed the wrong button, guys. All right, so let me just show that picture again. And I'm just going to use a still picture here to get it up. Oh, my poor mouse is going berserk. Hang on, guys. I'm having a battery problem.
So while you're fixing that, Sal, this might be a little bit off of topic, but, I'm just kind of thinking if, proteins had to develop independently, since we don't have, a like common ancestor for proteins, then and proteins are incredibly complex. And so we would need many tries, right? So it seems to me that if that's true, there would have to be like many non-functional proteins in our cells, right? So that we could even get the chances, and then they would need so many like additions in order to become functional that it'd probably take quite a long time to get those. So they'd be floating around for a long time, non-functional. Do we have non-functional proteins in our cells?
Yes, and we call those birth defects. We happen to know that maybe about 1,000 genes in the human genome are causing birth defects. And so that's the problem of kind of all or nothing functionality. And that's why I try to emphasize geometry, that it's just like parts of a car. You can't randomly change the geometry without breaking it. So if you're trying to make it evolve, like say a nut into a bolt, it doesn't work so well. Natural selection, or what they call natural selection, would actually prevent the evolution of new protein forms. It'll probably allow the evolution of like a little tweak of the same thing, but to actually make something work very differently than the way it is going to be challenging. So you point out something that's a problem I've seen for a while. To have all the trials and experiments that they're talking about, it's like if we do the accounting, you're going to be spending so much time just trying to explore all the probability space if it's random. And of course, they'll say natural selection solves that. And I say, no, this is exactly why I'm having discussions like this. You could see that the intermediates would be useless, in fact, possibly harmful to the system.
Okay, well, let me just say what I think the response would be. Well, you said natural selection, so maybe they would say, well, by now, after we've had all these millions of years of evolution, you see, we've weeded out all those non-functional proteins so that everything is nice and neat and functional now. So what's the response there?
The difficulty is, how do you know that you're going to get one? I'm trying to show the difficulty even getting just one. I mean, it's not just getting it to target the DNA. You have to have it with the right affinity. And then, like I said, you can't just have it just attached there and just be like a piece of useless glue. You have to modify the other parts of it so that it connects to the other things. And I haven't even gotten to chromatin modifiers. If you could show this here, I'm gonna just show some of the complexity of how zinc fingers are used. If you could show this still diagram.
Oh yeah, sorry. Okay.
So the stretch of DNA that this zinc finger targets is like 12 nucleotides long. Something like that, 12 to 15. And it's spelled one way, and then the reverse complement, 200,000. So this is like 200,000 bases, roughly, long. And then 200,000 bases later, there's another one that is reverse complement. This is like having parentheses when you're doing coding. For computer coders, you have to have punctuation marks. You have to have a left brace and a right brace. This is like putting a left brace here and a right brace here, and you're going to make something like this here. And okay, so you have to have this word right here, and then a word right here, and then you have the zinc finger, the CTCF, that's going to help put this cohesin complex right there. So you have to mutate the DNA, and you have bazillion years, and then you get another one here to get you the right one that corresponds. It's like your punctuation mark. It's a little bit more complex than that because you can have multiple different loop combinations. So I'm going to take this down, but then I'm going to show what this really leads to, and this is where it's really cool, because these loop domains, and let me share this. See if I could get this. Okay, how do I get this? I totally did something I didn't want to do. But I'll show it anyway. I don't know why I don't have the full screen anymore here. Anyway, sorry for the small screen. So I showed the extrusion loops. What these loops do is it ends up forming scaffolds for molecular machines. So let me show. Okay, so I'm sorry this is really tiny, and I don't know why my PowerPoint's just kind of blowing up here. Let me try to see if I could remedy that. Okay, from current slide. Praise the Lord. Okay, so you saw that loop. It has to be the right length. And this is basically like a robotic factory. And the robotic factory puts all sorts of little computational, it handles the gene regulation. And that's what the loops help do. It does more than that. And not only that, it's kind of hard to see this in the diagram because it's not zoomed in, but you can actually, this is called transchromosomal regulation, where you have a loop in one gene, and it contributes to constructing a factory here. You have different chromosomes, parts of different chromosomes aggregating together to create kind of a regulatory factory here. They actually may call that transcription factories, and it may involve different chromosomes. I mean, the amount of foresight required to do this is just incredible. Here's a better one. And this one involves link RNAs that come from one chromosome, and then this one connects the X chromosome, the chromosome 2, chromosome 15, chromosome 17, and this is where you're getting the interaction of these zinc fingers creating these loops, and then the loops interact to form a factory. And this is like, I'm just trying to say, you know, you have to think through just how difficult it is to stand up a system like this, the foresight required, because not only do you want to have the DNA somewhere, you want to have the DNA in the right positions and have the foresight to make a transcription factory. These transcription factories are different depending the cell type. So you have different transcription factories in the cells that, like, say, make your kidney cells, different ones that make your blood cells, your nerve cells, etc. Yeah, this is the amount of foresight is just incredible. So now let me move on a little bit. So that's just from the physical standpoint as far as robotic arms. So think of those extrusion loops. They can function as robotic arms, but they also function at what they call insulation, and that's another topic. This is an active thing that's being studied in the National Institutes of Health, one of my favorite places. Now there's another difficulty here if you're having zinc finger proteins. There has to be an orchestrated action in the chromosome to make the zinc finger even able to dock with the DNA, and that's chromatin remodeling. So going from left to right, you have the chromosome, and we're just going to zoom in a little bit, and you can see, like, say, you want to look a little piece of the arm, that's what it looks like. And if you zoom in, it has these, you could see this more of this 300 nanometer scale, and then the chromatin fiber. So you take that fiber and you zoom in, it looks like that. And then you take this and you zoom in, it looks like that. And finally you get down to the DNA. So this wire here is the DNA, and the little spools there are called histones. The reason I'm pointing that out is this, there have to be machines that will move these spools back and, you know, kind of stretch them out in the right position so that you'll have an exposed portion where the DNA is just hanging out like that. Then the zinc finger can, let me see, then the zinc finger can dock in that DNA. This has to be orchestrated so that where the zinc finger is going to land, it has to be spaced out just right. And this is dynamic because the spools will slide and move. And to do that, it's called chromatin remodeling. And there's a computation that involves these histones. So these spools here actually have computational tails here. I'm sorry. And this is like the tail. The tail is made of amino acids. So like there's like an arginine amino acid in position 2, a threonine in position 3, a lysine in position 4, et cetera, et cetera. And they'll have little chemical modifiers that are like flipping bits, and then you have reading machines that will read those bits, and that'll help orchestrate how these things will slide back and forth. So this is like multiple reading write heads and sliding things in position, and then it's going to open the way for the zinc finger transcription factors to come in. So a lot has to happen, and you have these machines called chromatin remodelers that help do this, these readers and writers, and you have these really monstrous, the machines that do this molecular wise are very, very complex, like this SWI-SNF. We can see when we use like fluorescent stains to see the state of the chromatin over the life of the cell. For example, we took the chromatin of 3-year-old twins on the left, and I think it's the same twins 50 years later, they took it, and you can kind of see all the computation going on with the light bells and whistles and lights, so to speak. We're able to actually see the changes. Also, in terms of like the embryo, we could see this chromatin computer changing states as it's developing into, this is going to be a nematode worm. And each of the little cells now are starting to differentiate. So like the ones in red are going to be the sex reproduction cells. And then the other ones will have different properties. So the zinc finger system has to be incorporated into all of this. It's more than just getting it to bind. It has to be orchestrated to bind in the right time, the right location, in the right cell type. It's just overwhelming. And there was a paper called Chromatin Computation. So I think I'm almost done. There was one part I wanted to point out. Yeah. So I was arguing, consider you have the FedEx truck that delivers packages. Let's say you get most of the digits correct. Like say the zip code of an address. Well, 12192 will be up here in New York, but 32192 will be down here in Florida. So even if you're asking Rebekah about millions of years, it's like, no, you don't get warmer when it doesn't tell you that you're getting closer and closer to the right location of DNA, because the spelling might be similar, but geographically, in terms of the location on the genome, it could be millions of base pairs away. So there's not going to be any natural selection that's going to say, oh, I'm going to, I just modify the zinc finger and I'm going to get closer and closer to where I need to be. No, you could be, you can be getting the right spelling. You're getting closer to the right spelling, but you're getting all different places in the genome. So like zip code 32192 is way out here in the middle. So even though they're spelled similarly, your zinc finger is just going to jump all over the place. So what was my point in showing this? When I was arguing with the evolutionary biologist that said, oh, we just do a phylogenetic reconstruction, that's really easy. I said, no dice, guys. You might be able to build your phylogenetic diagram, but that doesn't explain the difficulties. We can even assume common ancestry from a common zinc finger, but you don't resolve the problems of what I call the Federal Express problem. So that's pretty much an introduction to the zinc finger. And I think, if I may only horrify you more, I think I just scratched the tip of the iceberg as far as the complexity of all that's required. So thank you. I can't believe I got through this in an hour.
Good job. Michlas, do you have any final questions or comments?
No, not so much. As I said beforehand, I will have to take this and digest it and probably look a lot of it up because, well, being a layman, someone in the chat actually asked me what my position is, and no, as a basic, I don't agree with the young Earth creationism. I do accept evolution. I'm an atheist, so just to give my position here, I'm here to listen to what Sal has to say and his definition of this, so I can understand it, and in my opinion, to improve communication as much as we can, because I think the only way we can actually get anywhere is by being honest in these things. So while I strongly disagree for various reasons, I think it's only fair to listen why Sal thinks what he thinks about these things and let him explain these things, because I've seen people's comments out there, but one thing is for sure, Sal does know a lot more about this than I do. So it's, yeah.
Well,
yes.
I want to commend you all. You got about 3 semesters of graduate level biology in 1 hour thrown at you.
Well, I didn't really understand I didn't understand a quarter of it. So I'm not going to take credit for those semesters, but I appreciate you giving us the information.
Well, I just wanted to say that if the only takeaway is that there's a lot more that has to be accounted for, I've tried to show the parts that have to be accounted for if you're trying to evolve a system that involves a zinc finger. It's not. It's a lot more than just comparing sequences and looking at, oh, it has it in a monkey or a human. So what I'll even argue is I'm going to grant that there's common ancestry. Would that common ancestry require still statistical miracles to get the machinery to work? So you look at the phylogenetic diagrams, it's like, yeah, that looks reasonable from, if you want to build a tree, but it's a lot more than just building trees, these phylogenetic trees, and that's what yeah.
Oh yeah, Rebecca got it. I was about to mention that one.
Okay, so Design Disquisitions has this question. What are the latest non-design attempts to account for zinc finger proteins, and why do you think they don't succeed?
Oh, that is an excellent question. The non-design attempts are exactly what I showed with those phylogenetic diagrams. They assume that it evolved. They'll show the similarity and say, well, that shows, therefore we have a progression from one form to another. It's an easy step. And it's like, well, yeah, you could just say, well, there's changes in spelling and it looks superficially easy. So those are the non-designed ones. And so the term that I see a lot is coevolution. So you saw it like with the CTCF binding sites in the extrusion loops. You'd have to co-evolve the DNA, and you'd have to co-evolve the zinc finger. We have a lot of that. How do you have it? And they'll just say hey, we discovered coevolution. Now, when I gave this talk to the faculty that are more friendly to my position, they just laughed. I said, look, they're treating this like they discovered something amazing, how it's like evolution works more amazingly than we thought, that it could co-evolve the DNA and the zinc finger that targets that DNA. And so so they're out there, but when I shared it with my colleagues, they just laughed, because some of them are pretty savvy. I mean, these are deans of universities and researchers at the NIH, and they just laughed. I'm sorry to be a little harsh, but they realized this. The reason we know it, and Fuzz Rana who was on my channel, he said the reason we're starting to appreciate the improbabilities of biological systems forming naturally is we're trying to build pharmaceutical, medical technologies by kind of redoing being able to make molecules of our own to interact with the cell at this level of detail, and it's brutally difficult. We're going to throw our best minds at this and use design, our intelligent design, to match what we think naturally evolved. We think naively, well, if nature evolved it can't be that hard, and we're finding out the opposite. But so there are papers that try to address this, and they're usually just assertions of a foregone conclusion. They actually never work through the probabilities that I'm trying to describe here, where I talk about that they don't account for binding affinities, and they don't account for like those promiscuous domains that have to connect to this. They don't account for chromatin remodeling. They don't account for like when you have transchromosomal transcription factories. It's just like, there's you can just do these phylogenetic diagrams, and that's the bread and butter of evolutionary biologists, but then they ignore all the other problems, and that's the angle I'm coming from. But so there are papers out there, I've read them, and then I pass them on to our colleagues, because when they need a good laugh for the day. I'm sorry to be harsh, but that was their honest reaction.
Yeah, well, that's what frustrates me, is that no matter what we find, how difficult it is, the response by evolutionists is not doubt about their idea. It's not, oh, wait, maybe this couldn't have happened. It's, wow, look how amazing. Evolution is even more amazing than we thought it was. Okay. And for those who like to say that what we're doing is God of the gaps, whatever we don't understand, you just, put in God. Well, no, it's actually that we're recognizing the difficulties of these things. And we're saying, look, it's what we do know that's leading us to that conclusion. And if you're doing just because we haven't figured it out yet doesn't mean we won't, then you're doing science of the gaps. That means wherever there's a gap, instead of trying to come to what conclusion is most reasonable, you're just saying, well, science will figure it out. That is science of the gaps. And so I feel like no matter what, there's never a questioning of this theory by the people who believe it. So it doesn't really matter what we discover. So do you think, Michlas, do you think that's a unfair characterization?
Well, that's pretty hard for me to say, simply because I don't come at it from your side, so I don't experience this in the way that you say it, to be honest. I experience it a lot as a question of probabilities, and we've already talked about why I have another opinion on probabilities than most do. And I also come at it from the point that even if I became an expert in, let's just say, this topic, there would always be 100 topics that I would not be an expert on. And those cases, I would have to either start becoming an expert, which would be an impossible infinite task, or I would have to rely on the consensus of the majority of people. And I know this is, what should I say, discouraging, of course. But again, that is also why I'm here and trying to listen to the arguments and then see, okay, does these make sense to me? But I can't just listen to them like this and say, oh yeah, that's just evolution out the drain, or the other way around for the matter, because I would have to have time to actually think about these, go out, ask, try to find some professors on my school or other experts and say, okay, so if you look at this, what is your immediate assertion about this? Is there something you think I should look at this? For instance, right now we have Jay in the chat saying that the experts should debate this with Sal. And I would be interested to hear what experts have to say in this case. But I would absolutely be.
They have
and sorry, it is Jay, the name.
Yeah, they have debated me in text, and I dropped out of that forum. That's the skeptical zone. And Rebekah, I copied you a link to that.
Oh, okay.
It doesn't necessarily represent my present views, that interaction, but you could see that I went up against the best there, including Joe Felsenstein. And he's considered the master of evolution. And so the problem is we're talking past each other a lot. As I said, evolutionary biologists will say, look at these phylogenetic nested hierarchical trees. And they'll say that that's proof. And I'll say, I'm going to grant you have common descent in these nested phylogenetic trees. We have people like Michael Behe who accept common descent, but will say that it's another layer of probability beyond common descent. It's the intricacy of actually having all the parts conspire to make these complex systems. And so they're thinking that it's adequate enough to just establish like similarity, build phylogenetic trees, argue for common ancestry, that solves the problem of complexity. And I'll point to Michael Behe. He says, I accept all that, but you still haven't explained the probabilities. So we can accept common ancestry, but it would seem to him that all these variations, and he had something called front-loaded evolution, where the evolution's in a process of common descent, the mutations would have to be kind of like had foresight or divine guidance or something like that. He called it front-loaded evolution. He only had 2 pages in one of his books, Darwin's Black Box, that mentioned it. He never developed a theory. He said, that's not really my worry. He said, if you ask me, that's what I think. It's not a well-developed theory. So when you see that debate, you'll see a lot of us talking past each other. They're talking these phylogenetic trees and the nested hierarchies. I'm going to grant all that. It would seem to me you would still need divine guidance or some sort of intelligent guidance, as Michael Behe's asserting, or what he would call front-loaded evolution, where, this is all, the first cell kind of had all the capacity to kind of evolve into all the things we have today. It's not a very well-developed theory, and he's never pursued it. So I just wanted to say thank you that you wanted to see me interact with the experts, and I'm happy to. I'm really happy to talk. That's why I have Dr. Dan on my channel a lot. I mean, not a lot, but I'm happy to have discussions like this. And what he said, well, Sal, the reason we admire you is you get into the nuts and bolts, and you have described the complexity of some of the systems correctly. And I have this on video. Dr. Dan said, I'm just not there to invoke a supernatural cause. And and I thought that was one of the best interactions I've had with Dr. Dan. And I consider him a friend. I've taken flack because I'm a creationist. He's not. Creationists have hated me for being chummy with him. I'm just like, well, I like the guy and I love interacting with him. So there have been discussions between me and, professors in secular schools who are evolutionary biologists. Check out my channel. You see Dr. Dan on my channel. You can see us interacting.
Check out Evidence and Reasons. And I think we have to close it up now because it's quite late for Michlas. Did you want to say something?
Yeah, I actually have one question because now you mentioned it so many times in this little talk. So I have to ask, when you say the probabilities, I know the exact probabilities haven't been calculated and probably never will be calculated. But what order of probabilities do you think we are talking about? Or talking like it's one against 100 coin flips, basically? Or where are we in your opinion?
I would think they're astronomical. So the individual zinc fingers, not so much, like say on the order of a few 100,000. You can just take that from you need an alphabetic string of 28, and let's say you need about 10 in the right position, so like say 20 to the 10th power or something like that. So it's something small. But the problem is when you have multiple zinc fingers, then it becomes more problematic. But then when you consider, you can't just stop with a zinc finger. It has to be attached to like a crab component, another domain. And then the crab component has to attach to the CAP1 protein plus the MAGE. And you need all these spellings both in those proteins. So you have to co-evolve, all these pieces to have these functioning systems. And so the individual probabilities may not be that insurmountable. It's like I could flip a coin, high probability of getting heads. But when you have to flip like 100 coins and have them all heads, it starts to become, the probabilities, improbabilities multiply. So I view it as astronomical. And so, the easiest way to calculate the probabilities are with abiogenesis. So for me, I'm just saying for myself, those are very easy to calculate relative to these things because we're dealing with simpler systems. And if that's astronomical, and I believe a miracle made it, then I can believe, when I'm confronted with this, even if I don't know the exact numbers, I'm a little bit more inclined to accept the miracle. And so it's not as rigorous. But if you wanted, that's my intuitive feeling about this. It would be on the astronomical level.
Yeah. Okay. Well, thank you. Bye.
Yep. Thank you so much, Sal. Please subscribe to Sal's channel, Evidence and Reasons, and God bless you guys. Have a good day.